Because this lesson is specifically on linked genes, we will be discussing mapping the distance between genes using recombination data, also known as gene mapping. We will go over recombinants, linkage maps, and cytogenetic maps.
In an earlier chapter, we studied Gregor Mendel's genetic experiment involving pea plants. We learned what happened when he mated pea plants with different heights, seed colors, seed shapes, and flower color in a monohybrid cross. When Mendel later looked at two different characteristics at a time, he realized that some offspring looked like the parents and some did not. We know these as dihybrid crosses. We can look at a test cross between a pea plant with yellow round seeds that is heterozygous for both seed color and seed shape, or big Y, big R, little r, and a pea plant with green wrinkled seeds that is homozygous for both recessive alleles, or little y, little r, little r. On the top of the Punnett square are all the possible gametes that the yellow round parent can give to its offspring. On the left, the green wrinkled parent can only give one combination of gametes to the offspring since it's recessive for both traits. When the two parents cross, half of the offspring look identical to one of the parents. However, the other half of the offspring do not. These offspring are known as recombinants since they do not possess both characteristics that either parents have. In other words, the recombinants that formed were a yellow wrinkled pea plant and a green round pea plant. Neither of these two offspring look like the parents. After countless experiments, it was concluded that recombinant offspring were formed due to the crossing over that occurs when homologous chromosomes are paired during prophase of meiosis I. In gene mapping, we focus primarily on these recombinant offspring that form.
After learning about linked genes and recombination due to crossing over, one of Morgan's students, Sridhar, discovered genetic maps, which are an ordered list of genetic loci along a particular chromosome. He hypothesized that the recombination frequency or percent of recombinant offspring all depend on the distance between genes on a chromosome. So on this chromosome, what that's saying is all of these genes have a different distance between them. He believed that crossing over approximately was equal at all points along the chromosome. He said that the further apart two genes are, the higher the probability that a crossover will occur between them, and therefore the higher the recombination frequency. So what that means is these two are less likely to cross over than these two. One type of genetic map is a linkage map. A linkage map is based on recombination frequencies. Sridhar conducted an experiment to prove these. He concluded that linkage maps are based on assumptions that the probability of a cross between two genetic loci is proportional to the distance separating the loci. So the distance between the genes of a linkage map are known as map units. It's pretty simple. One map unit is equal to 1% recombination frequency. Now down below we have a chromosome that has 3 different genes. As it says, the first two have 2 map units in distance, so that's just equal to 2%. And the next one has 4 map units, which is just equal to a 4% recombination frequency. These recombination frequencies make up the linkage map for one specific chromosome. Now these genes are arranged on the chromosome in the order that best fits the data. Sridhar observed recombination frequencies between 3 Drosophila gene pairs, which were B for body color, CN for cinnabar or eye color, and VG for wing size. Because of the recombination frequencies of 9% between B and CN, and 9.5% between CN and VG, he concluded that CN would be found halfway between B and VG. He then concluded that the crossovers between CN and B and CN and VG were half as frequent as the crossover between B and VG. You might be asking yourself why it doesn't equal 18.5% because This 9 and 9.5 would equal to 18.5 map units, or 18.5%. That is because the few times that one crossover occurs between B and CN, and another crossover occurs between CN and VG, the second crossover would cancel out the first, reducing the observed recombination frequency while contributing to the frequency between each of the closer pairs of genes. So the value of 18.5%, or 18.5 map units, is closer to the actual distance between the genes.
While these linkage map frequency rules seem simple, there are some exceptions. Interpreting the recombination data is a little bit more complex. As we recall from earlier, the further apart the genes are on the chromosome, the more likely they are to become recombinants than the ones that are closer to each other, which are less likely. This means that they are more likely to cross over during prophase 1 of meiosis and create recombinant chromatids. Sometimes genes on a chromosome are so far from each other that a crossover between them is practically certain. The actual maximum value for two genes, which are extremely far apart in terms of observed frequency recombination, is 50%. This maximum value, as in how far apart the chromosomes are, is hard to tell apart from genes on different chromosomes. This means that genes on the same chromosome, which have the max value of 50%, and the genes on different chromosomes will have indistinguishable results. Therefore, these genes on the same chromosome, although physically connected, are genetically unlinked. This means that the alleles assort independently, acting as if they were on different chromosomes. Two known genes for P characters that Mendel studied were actually on the same chromosome and assorted independently, but the distance was so great that the linkage is not observable in crosses. In finding the distance of genes located far apart, we can use genes closer to each other in between the further genes by adding those frequencies together. Using recombination data, Sridhar and his colleagues were able to map numerous Drosophila genes in linear arrays. They found that the genes clustered into 4 groups of linked genes, or linkage groups. As you can see in this figure, the number at each gene locus indicates the number of map units between that locus and the locus for the appendages on the head, which is on the left, also known as the distance from the aristae. Also, it can take note that more than one gene can affect a given phenotypic characteristics, such as eye color. From these linkage maps, we are also able to see the Drosophila chromosomes, and we can see that in contrast to the homologous autosomes, the X and Y sex chromosomes have distinct shapes. In turn, this proves additional evidence that genes are located on chromosomes and that each chromosome has a linear array of specific gene loci. However, linkage maps do not take note of the real distance. That is why it is important to learn about cytogenic maps.
When Sridhar created his model of crossing over with Morgan, they assumed that the length of the gene corresponded directly with the recombination rate. Thus, when a certain gene set was determined experimentally to have 17% recombination rate, it was thought that the gene set covered 70% of the length of the chromosome. But the frequency of recombination is not directly relatable to the length of the gene set. Thus, where we might have a recombination rate of, say, 17% between these two genes, we might still have a recombination rate of 10% between these two genes, regardless of the fact that the distance between these two genes is a higher percentage of the overall chromosome length than the distance between these two genes. And so certain regions can be more or less prone to crossing over, regardless of the actual area or length that they have of the chromosome. And so Sridhar's map unit system of percentage recombination are not accurate in representing the gene distance as it relates to any unit. So you cannot take his map units and directly relate them to any direct unit of length, such as a nanometer or whatever it might be. So the linkage maps created by Sridhar were accurate to the order of the genes on the chromosome, but they were inaccurate, as we said earlier, to the distance between such genes. And so, cryogenetic maps, or karyotypes as they're more often known, were developed to accurately portray such distances. In a cytogenetic map, we can see the physical appearances of the actual chromosomes in the cell, and the chromosomes are extracted during the period of mitosis in the cell, usually during metaphase or prometaphase A, and treated with dyes to produce banding patterns at gene locations in the chromosomes, and ordered and sorted into chromosome type as we see here. In practice, we can see that the genetic order, as predicted, is on the karyotyped chromosomes, is consistent with what we can see in the linkage-mapped chromosomes, but the distances, based off of the banding patterns of the actual genes after being treated with the dyes, are completely different.